I want you to imagine a family somewhere in America right now.
Yeah.
A family that just hasn't slept properly in like 30 years. Think about the quiet that falls over their house every single year on one specific date.
Right, that agonizing anniversary.
Exactly. And, they don't talk about it at the dinner table anymore. They've stopped calling the local precinct because the detective who originally took their case retired 2 decades ago.
And then the guy who took over for him probably retired too.
Yeah,
Yeah.
exactly. But every year, they mark this quiet anniversary for a loved one, like a daughter who went out and never came home, or a mother found in her apartment with the person who did it just walking away, breathing free, living a completely normal life.
It's a devastating reality to sit with. And for decades, that was simply the grim, kind of immovable landscape of cold case homicides.
Yeah,
The bitterest part of it all, I think, was that the evidence was usually sitting right there in a locker.
like literally just in a box.
Exactly. The DNA was physically present. But the methodology to actually read that evidence and connect it to a human being, it just hadn't caught up yet.
Until right now. So, welcome to today's deep dive. We are looking at a comprehensive breakdown of the DNA revolution.
It really is a revolution.
It is. It's this scientific seismic shift that is tearing open America's and really the world's most impossible cold cases.
Yeah. And our mission today is to understand exactly how a single strand of DNA is suddenly breaking decades of absolute silence.
Right. We're going to explore the new scientific methods that are rewriting the rules of criminal justice, and of course, trace the profoundly human stories behind these headlines.
Because there's a lot of them lately.
There are. And before we get into the deep mechanics of the science, I just want to say we're pulling from a massive stack of sources today, and the through line is just staggering.
It really is.
We're going to lay out a roadmap for you so you know exactly where we're headed. The key findings from the solved cases we're covering today show how the notorious Golden State Killer was finally caught after a 44-year reign of terror.
That's a huge one.
Huge. We'll also look at how a 49-year-old murder in California, the Karen Percifield case, and a 26-year-old murder in Minnesota, Jeannie Childs, were solved using distant relatives' DNA.
Right, through genetic genealogy. And we'll also examine how the Florida Keys managed to crack 3 separate gender mysteries in a single year.
Which is wild.
It's completely wild, ranging from an 8-year-old hurricane victim to a 38-year-old cold case. Plus, we actually have a breakthrough from just May of 2026 in Germany, which led to a father's arrest for a crime committed 25 years ago.
So the sheer variety of these cases, from serial killers to single tragic homicides to environmental accidents, it just shows the breathtaking scope of what has happened.
Yeah, the scale is massive.
Okay, so let's unpack this. Because the most mind-bending part of all this is that the clues weren't missing. Like you said, the DNA was preserved. It was waiting.
Right, it was right there.
The investigators essentially had the answer key locked in an evidence vault. They just didn't have the light to read it by.
Yeah, and if we connect this to the bigger picture, this is exactly why you, the listener, need to care about this topic.
Yeah.
The timeline of justice has been completely upended.
Totally.
We used to think of justice as having a ticking clock, right? If you didn't catch a suspect in the first 48 hours, the odds just plummeted.
The trail goes cold.
Exactly. If you didn't catch them in a year, you probably never would. But now, the definition of a perfect crime is rapidly going extinct. Time is no longer the ultimate shield for a perpetrator.
Which is a crazy concept to wrap your head around, and it means we need to talk about the scientific pivot that actually made this possible.
All right, the turning point.
Because for the longest time, the criminal justice system asked a DNA sample one very specific question, which was, who is this? But the revolution happened when they started asking a completely different question, who is their cousin?
That's the paradigm shift right there.
So we need to look at the flaw in the old system first, which relied on something called CODIS. Let's start there.
Right, so to understand the flaw, we have to look at how traditional forensic DNA testing actually works.
Yeah.
Law enforcement has been using this since like the late 1980s.
Okay.
It relies on analyzing what are called short tandem repeats, or STRs.
Here's TRs.
Right. When traditional labs extract DNA from a crime scene, they aren't looking at the entire human genome. They're just looking at roughly 20 very specific locations on the DNA strand.
Because looking at the whole genome back in the 80s and 90s was technologically impossible, right? Like, they had to find a shortcut.
Precisely. They found these areas in the DNA, the STRs, where short sequences of genetic code just repeat themselves over and over. And the number of times they repeat varies wildly from person to person. So at location A, your DNA might repeat 5 times, but mine repeats 12 times. At location B, yours repeats 8 times, mine repeats 3 times. When you look at 20 of these locations, the combination of those repetitions is statistically unique to you.
So it's basically just a genetic barcode.
Yes, exactly, a barcode.
Right.
And once law enforcement gets that barcode from a crime scene, they store it in the FBI's database, CODIS.
The combined DNA index system.
Right. And the logic is pretty straightforward. A killer commits a crime, leaves their DNA behind, and then maybe years later gets arrested for something completely unrelated.
Like a burglary or something.
Exactly. Their profile gets entered into CODIS, the system pings, and suddenly this new arrest is connected to the old crime scene.
That makes sense.
That is the theory, anyway. And for many cases, I mean, it has been wildly successful.
Sure.
But there is a massive blind spot built into the very architecture of that system.
Right. And reading through the sources, it feels like CODIS is basically a really strict VIP club.
That's a good way to put it.
Yeah, like it is highly effective at tracking you if you're already on the guest list. But if the suspect is a first-time offender, or if they committed one horrific act and then lived a quiet life and were never arrested for anything else,
Then they aren't on the list.
they aren't on the list. They are totally invisible to the system.
And that blind spot is the exact reason why hundreds of thousands of cases just hit a brick wall. The detectives did everything right. They secured the scene, they collected the DNA, they built the STR profile,
Yeah.
they ran it through CODIS, and the screen just returned a blank result. No match. The database simply had nothing to compare it against.
So you have the complete answer right in your hand, but you literally don't have the dictionary to translate it.
Exactly.
That is where forensic genetic genealogy steps in, or FGG. This is the core paradigm shift of our deep dive today. Instead of needing the killer's direct DNA to be in a criminal database, you just need to find their relatives in a public one.
Right. And this requires a totally different type of DNA testing. We move away from those 20 STR markers.
Yep.
Instead, the laboratories use advanced sequencing to look at single nucleotide polymorphisms, or SNPs.
SNPs. Let's break that down for the listener. What is the actual mechanical difference between an STR and an SNP?
Okay, think of the human genome as a massive encyclopedia containing billions of letters.
Okay, tracking.
Traditional STR testing is like skimming the encyclopedia and just counting how many times the word "the" appears on 20 specific pages. It gives you a rough barcode.
Right.
SNP testing, on the other hand, is like reading the actual spelling of hundreds of thousands of individual letters across the entire encyclopedia. You're looking for very specific known typos or variations that are shared among family members.
Wow. Okay, so it's exponentially more detailed.
Exponentially.
And because it's looking at those shared familial variations, it taps into the basic math of human genetics. Like, we know siblings share roughly 50% of their DNA. First cousins share about 12.5%.
Right.
But the sources point out that this method tracks relationships much further out. Even third cousins share a measurable, identifiable amount of DNA. It's roughly 0.78%.
And that tiny fraction, that 0.78%, is the crucial thread. A third cousin is someone who shares a set of great-great-grandparents with you.
Wow.
Yeah. And over the last decade or so, commercial genealogy databases like GEDmatch, Family Tree DNA, and MyHeritage just exploded in popularity. People were buying these DNA kits for the holidays.
Oh, yeah. Everyone was getting them.
Right. Millions of people uploaded their SMP profiles.
Just to find out where their ancestors came from, or, to see if they have a long-lost aunt in Ireland or something. The irony here is so profound to me.
It really is.
Investigators realized that millions of people who were just innocently uploading their DNA to research their family history were inadvertently weaving this massive, inescapable net to catch criminals.
They voluntarily built a global kinship database.
Yeah.
If a killer's distant cousin, someone they have never even met, perhaps living on the other side of the country, uploads their DNA out of pure curiosity, that cousin becomes the anchor point.
It's incredible. And our sources lay out a 6-step process for how this reverse genealogy method actually unfolds in practice. It's a fascinating journey.
It really is a step-by-step grind.
So step 1 is getting that complete genetic profile using advanced sequencing from the crime scene evidence.
Right. And once you have that incredibly detailed SMP profile, step 2 is uploading it to a public genealogy database, like GEDmatch. The investigators are essentially querying the database, asking the algorithm, who in this system shares genetic variations with our crime scene sample?
Which brings us to step 3. The database spits back a list of potential relatives.
Yeah.
And it is almost never the killer themselves, right? It's usually a list of distant connections, like a third cousin twice removed.
Exactly. And this is where the true investigative labor begins. Step 4 is the domain of the forensic genealogists.
Right.
It's important to realize this is not a computer automatically spitting out a suspect's name. The genealogists have to build a massive family tree, working backward from that distant cousin match. Yep. They dig through historical birth records, century-old census data, marriage licenses, obituaries, and modern social media. They are painstakingly mapping out an entire family network over multiple generations.
I really want to pause on that because the mental gymnastics required are wild. They are building a family tree for someone else without knowing who their ultimate target is.
Right.
They trace branches all the way up to great-great-grandparents in the 1800s, and then they have to turn around and trace every single descendant back down to the present day.
It results in a tree with thousands of individuals, which leads to step 5, narrowing the field.
Filtering it down.
Exactly. The investigators take this massive tree and apply filters. They look at the age of the individuals, cross-referencing who was alive at a certain age at the time of the crime.
Okay.
They look at geographic locations, who live near the crime scene. They apply physical descriptions if they have them. They eliminate branches of the family one by one until they are left with a handful of names, or sometimes just a single individual.
But the genealogy alone doesn't put someone in handcuffs. It only provides a lead.
Correct.
That brings us to the final hurdle, step 6. The police have to conduct physical surveillance on this final suspect. They wait for the suspect to discard something out in the open that contains their DNA, like a coffee cup, a tissue, a cigarette butt.
Yeah.
They swoop in, collect it, run a traditional STR test on it to compare against the original crime scene DNA and confirm the match.
It is deeply methodical. But this entire 6-step pipeline falls apart if you can't achieve step 1.
Getting the profile.
Right. You have to be able to extract a viable, high-quality genetic profile from crime scene evidence. And when we're talking about cold cases, that evidence might be 50 years old.
Yeah, that brings us to the lab that is really changing the entire landscape. Our sources specifically highlight Othram Inc.
Ah, yes, Othram.
Based in the Woodlands, Texas. They are the engine behind nearly every major breakthrough we are discussing today.
To appreciate what Othram does, we have to look at the severe limitations of traditional forensic science. Evidence from the 1970s or 1980s wasn't stored in pristine, climate-controlled environments initially.
Oh, I'm sure it wasn't.
It was exposed to fluctuating temperatures, humidity, bacteria, and ultraviolet light. All of those environmental factors physically break down the DNA molecule. They shatter the DNA backbone into microscopic fragments.
So for decades, if a detective handed a traditional lab a blood smear from 1978 or, a single hair root from 1983, the lab would just shake their head. They would say it was scientifically useless.
Because the DNA was too fragmented to read those 20 STR markers.
Right. But Othram developed a process they call forensic-grade genome sequencing.
Yes.
They don't rely on finding those intact STR regions. They use incredibly sophisticated sequencing machines that can read the shattered, tiny fragments of DNA.
And then they use advanced bioinformatics, basically massive computational power, to stitch those fragments back together based on overlapping sequences.
It's like finding a vital document that has been put through a cross-cut shredder, soaked in water, and left in the sun until the ink bled.
That's a great analogy.
Right. A traditional lab looks at it and says, I can't read the sentences. Othram looks at it, scans every microscopic piece of paper, uses a supercomputer to match the torn edges and the bled ink, and reconstructs the entire page.
That mechanism of overcoming degradation is the technological leap that opened the floodgates. Once they proved they could resurrect dead evidence, law enforcement realized they possessed a completely new weapon.
Which brings us to the case that proved it to the world. We are moving from the theoretical science to the historical turning point. The apprehension of the Golden State Killer.
The context of this case is absolutely essential to understanding the magnitude of the breakthrough. This wasn't just a cold case. This was a generational trauma for the state of California.
And huge.
The reign of terror lasted from 1974 to 1986.
And the facts are chilling. Over that 12-year period, this single individual committed at least 13 murders, over 50 sexual assaults, and more than 100 home burglaries.
Yeah.
And his geography was massive. He started in the Sacramento area in Northern California, where he was known as the East Area Rapist, and eventually moved all the way down to Orange County in Southern California, evolving into the original Night Stalker.
The psychological terror he inflicted just cannot be overstated. He didn't just attack, he orchestrated nightmares. The sources detail how terrifyingly methodical he was.
Oh, it's awful.
He would surveil target neighborhoods for weeks, learning the routines of the residents, mapping the escape routes, and figuring out who lived where. He would enter homes through unlocked windows or sliding glass doors in the dead of night.
His methods inside the house were pure psychological torture. He would use the victim's own shoelaces to bind them.
Yeah.
And one of the most terrifying details mentioned in the sources was his use of dishes.
Right, the dishes.
He would stack plates and teacups on the backs of male victims while they were bound on the floor. He would whisper to them that if he heard a single dish rattle or fall, he would kill everyone in the house. It completely paralyzed them with fear while he assaulted their partners.
He was also highly disciplined in his forensic countermeasures, which was really unusual for the era.
Yeah, he knew what he was doing.
He wore gloves to avoid leaving fingerprints. He often brought his own supplies, like pre-cut bindings, to the crime scenes. He monitored police scanners to track law enforcement movements in real time.
Wow.
And perhaps most sadistically, he would make taunting phone calls to his surviving victims months or even years later, just to remind them he was still out there, still watching.
That is just terrifying. Detectives across multiple jurisdictions in California eventually realized they were hunting the same phantom. As DNA testing emerged in the 1990s, they were able to link the Northern California assaults to the Southern California murders.
They had his DNA barcode.
But they hit the code as blind spot. They had the DNA, but absolutely no name to attach to it.
And that phantom status remained for decades. By the 2000s and 2010s, this case had achieved a mythical status in law enforcement circles. Two key figures really kept the pressure on during those long, cold years.
Right.
First, retired investigator Paul Holes, who dedicated a massive portion of his career to hunting this man, meticulously preserving the evidence. And second, the author Michelle McNamara.
Michelle McNamara is actually the one who coined the moniker Golden State Killer in a 2013 article. She spent years investigating the case with absolute obsession, eventually writing the phenomenal book I'll Be Gone in the Dark.
It's an amazing book.
It is. Tragically, she died in 2016 before the book was published and before the killer was caught. But her work brought the public's attention back to the case in a profound, unavoidable way.
Despite all that brilliant detective work and heightened public awareness, the breakthrough didn't come from a standard tip line. It came from the genealogy hunt. Yeah. In 2017, Paul Holes connected with Barbara Reventer. She's a retired patent attorney turned forensic genealogist who was a pioneer in what was then an incredibly experimental field.
They took the Golden State Killer's DNA profile and uploaded it to GED Match. At that time, GED Match had nearly 1 million users. When they ran the profile, the algorithm returned matches.
But as we discussed earlier, it wasn't a match to the killer. It was a list of distant relatives, third cousins.
Think about what that actually means for an investigator in 2017. I really want to emphasize this. They didn't have modern AI instantly mapping family trees.
No, not at all.
Ray Venter and a small team had to trace those third cousins back to the 1800s to find their common ancestor, and then trace every single descendant forward through time. This was excruciating manual labor.
Yeah, they were sitting at computers, sifting through century-old census records, birth certificates, death notices, and marriage licenses.
Just reading through old newspaper archives and scrolling through modern social media profiles to see who was related to who. The family tree they built grew to contain thousands of individuals.
Then slowly, they applied the filters. They needed a male of a certain age who had ties to the specific geographic locations in California during the times of the attacks. Thousands of names became hundreds. Hundreds became dozens.
Until finally, after months of relentless work, the massive tree narrowed down to one single branch, one family, and then one man.
Joseph James DeAngelo Jr.
A 72-year-old former police officer living a quiet, seemingly mundane life in Citrus Heights, a suburb of Sacramento.
The fact that he was a former police officer suddenly made sense of his methodical countermeasures. He understood law enforcement tactics because he had been trained in them, but they still needed step 6. To confirm he was the monster they were looking for, they needed a direct DNA sample.
Right, so investigators quietly tracked DeAngelo. They managed to collect a discarded tissue he left in a public parking lot. They rushed it to the lab, ran it against the DNA left at the crime scenes decades earlier, and it was a perfect match.
On April 24, 2018, police arrested Joseph James DeAngelo in his driveway.
Wow.
What's fascinating here is the sheer banality of his arrest compared to the terror of his crimes. His immediate response, the quote he reportedly gave to the officers as they took him into custody was, "I have a caretaker."
I have a caretaker. It is so bizarre. He's this legendary boogeyman responsible for some of the worst crimes in California history. And in the moment of his capture, he tries to play the card of a frail, confused old man who needs help.
It speaks to the psychological disconnect of these offenders when the reality of their actions finally catches up to them. Ultimately, in June 2020, at the age of 74, D'Angelo pleaded guilty to 13 counts of first-degree murder and numerous rape charges, receiving a sentence of life in prison without parole.
But the legacy of this case wasn't just taking one serial killer off the street. It was the proof of concept. When Joseph James D'Angelo was identified, it was a catalyst for the entire global law enforcement community.
Exactly. Every detective looked at that headline, looked at the boxes of cold case files sitting in their own storage rooms, and realized they had a proven method.
The dam broke.
When that dam broke, the sheer volume of decades-old silence that was shattered is hard to comprehend. That brings us to two specific cases that demonstrate the deeply human toll of this waiting game. We move from a notorious serial killer to deeply personal tragedies. Let's look at the case of Karen Percifield.
Karen's case is a profound example of what happens when the science of today reaches back to rescue the victims of yesterday. The crime occurred in May of 1976 in Santa Cruz County, California. Karen was only 25 years old. She was found stabbed to death in Aptos Village Park.
The local deputies worked the case hard. They collected forensic evidence from the scene, pursued leads, and interviewed everyone they could. But in 1976, forensic DNA profiling as we know it did not exist.
Right.
Serology could tell them blood type, but that was about it. The trail went cold, and it remained cold for 49 years.
I really want you, the listener, to try and put yourself in the shoes of Karen's family. Imagine the psychological torture of waiting almost half a century for an answer.
It's just unthinkable.
You watch the original detectives retire. You know the physical evidence is sitting in a box somewhere, slowly aging. You watch the rest of the world move on. Technology advances, presidents change, entire decades roll by, and you are still emotionally stuck in May of 1976.
Every time the phone rings, for 49 years, you wonder if it's the police.
Exactly. It is an unimaginable burden. But in the fall of 2023, the Santa Cruz County Sheriff's Office took a pivotal step. They took that preserved evidence from the 1976 crime scene and submitted it to Othram.
Using that forensic-grade genome sequencing, Othram managed to pull a comprehensive DNA profile from evidence that was nearly half a century old and heavily degraded.
They ran the reverse genealogy playbook, and by April 2025, they were ready to make an announcement. The family tree had led them to a man named Richard Somerhalder. But this is where the story takes a deeply frustrating twist.
Investigators discovered that Richard Somerhalder was a multiple killer. In fact, just months after Karen Percifield was murdered in May of 1976, Somerhalder was arrested in September of 1976 for two other murders in the exact same county.
He was right there. He was in the system for other horrific crimes. But because they couldn't connect the forensic dots back then, he was never tied to Karen. And the ultimate tragedy here is that when they finally identified him in 2024 through genealogy, they realized Somerhalder had died in 1994.
This raises a profound philosophical question about the nature of justice. Richard Somerhalder will never sit in a courtroom for Karen's murder.
Right.
He will never face a judge or a jury. He will never have to look Karen's family in the eye and answer for what he did. Is a name alone enough? Can we call that justice when in trial is biologically impossible?
I want to push back on the idea that justice only happens in a courtroom. Based on what these families express in the sources, I think the answer is yes, a name is enough. It is a different kind of justice. It is the justice of truth.
Yeah, that makes sense.
For 49 years, Karen's family had a void, a terrifying question mark. Now they have a fact. They know who did it, and they know he can't hurt anyone else. That power of naming the killer is monumental. In fact, Othram solved 5 separate California cold cases in the single month of April 2025 alone. That's 5 families getting the truth back.
That pursuit of truth brings us to our next case, which introduces a whole new layer of complexity to the legal battlefield. As this technology expanded, it was inevitable that it would face intense scrutiny in the courts. We moved from California in 1976 to Minneapolis in 1993 and the murder of Jeannie Childs.
Jeannie was 40 years old when she was found stabbed to death in her apartment. Like Karen's case, the investigators collected DNA from the crime scene, but the killer wasn't in CODIS, so the case went stone cold for 26 years.
Until 2018. The same year the Golden State Killer breakthrough made headlines, investigators in Minneapolis reopened Jeannie's file. They sent the crime scene DNA to a lab called DNA Solutions, Inc. and had them create an S&P profile. They took that profile and uploaded it to MyHeritage.
And unlike the Golden State Killer case where they had to trace third cousins back to the 1800s, this search yielded an incredibly close hit. A first cousin.
That is a massive shortcut for the genealogists.
Definitely.
From a first cousin, they were able to build the family tree very quickly. It pointed squarely at a man named Jerry Arnold Westrom.
Westrom was a Minnesota businessman. He had no violent criminal record, which is exactly why he was completely invisible to the traditional CODIS system. But the police still needed step 6, the direct DNA confirmation.
They put Westrom under surveillance. Eventually, they watched him at a hockey game, eating a hot dog. He wiped his mouth with a napkin and tossed it in the trash. The police swooped in, collected the napkin, tested the DNA on it against the DNA from Jeannie Child's apartment, and it was a match.
But Westrom didn't just quietly plead guilty. She fought back hard, and his defense strategy challenged the very foundation of the DNA revolution. His legal team launched an intense assault on the entire methodology of forensic genetic genealogy.
Let's dive into that legal argument.
Wait, let's play devil's advocate for a second. If I spit my gum out on the sidewalk or toss a napkin in the trash, does law enforcement really have the right to just scoop that up, sequence my entire genetic code, and trace my entire family history without a warrant? Isn't that a massive privacy violation?
That is precisely the argument Westrom's offense made.
Yeah.
They argued that taking crime scene DNA and uploading it to consumer databases without a specific search warrant was a violation of the Fourth Amendment, which protects against unreasonable searches and seizures.
Right.
They essentially claimed that you have a reasonable expectation of privacy in your genetic code, and that the police were conducting a warrantless search of millions of innocent people's data just to find him.
It is a fascinating legal argument. Does the privacy of the millions of people in the genealogy database get violated when police search it without their explicit knowledge?
It became a landmark legal battle that went all the way up to the Minnesota Supreme Court. And the court's ruling was decisive. They affirmed that the process was lawful.
What was their reasoning? How did they justify it?
The court relied on a couple of key legal doctrines. First, regarding the consumer databases, they noted that users voluntarily upload their DNA to these platforms and agree to the terms of service, which often include provisions for law enforcement use.
So you kind of waive your rights there.
Exactly. You don't have a constitutional expectation of privacy for information you willingly share with a third party. Second, regarding the napkin, the court relied on the abandoned property doctrine. When you throw a napkin in a public trash can, you are legally abandoning it. You forfeit your expectation of privacy over that item.
So because of that Supreme Court ruling, Westrom's conviction for first-degree premeditated murder stood. He was sentenced to life in prison, and his conviction was finalized in 2025. It took 26 years, but Jeannie Childs got justice, and the legal framework for using genealogy in Minnesota was cemented.
That legal validation was crucial because it allowed investigators to keep pushing the boundaries without fear that their cases would be thrown out of court. But as we transition into the next phase of our deep dive, we have to acknowledge a profound shift in how this technology is used.
Oh, definitely.
DNA doesn't just catch killers. Sometimes its most profound purpose is simply giving a name back to someone who has been lost.
And this brings us to what I find to be one of the most remarkable segments in our sources, the Florida Keys cluster. We are shifting from solving murders to solving mysteries of identity.
Yeah.
Here's where it gets really interesting. The Monroe County Sheriff's Office, working with Othram and the Florida Department of Law Enforcement, managed to solve 3 incredibly diverse, decades-old John Doe cases in rapid succession. And this highlights the brutal battle between forensic science and the natural environment.
The environment in the Florida Keys is uniquely hostile to forensic evidence.
Sure.
You have intense heat, oppressive humidity, corrosive saltwater, and frequent hurricanes. From a biochemical standpoint, it is a biological shredder for DNA.
Wow.
Heat accelerates hydrolytic cleavage, which physically breaks the DNA strands. Microbes and bacteria in the swampy environments rapidly scavenge organic material. Yet the advanced sequencing technology prevailed. Let's look at Case 1, the tragedy of James Donald Schlake.
This one is heartbreaking because it's tied to a natural disaster. In September 2017, Hurricane Irma devastated the Keys. In the chaotic aftermath, amidst the immense debris on Big Pine Key, investigators found human remains in an advanced state of decomposition. They couldn't identify him.
And this wasn't a murder. The medical examiner eventually ruled the cause of death as an accidental drowning due to the hurricane. But for 8 years, his family had no idea what happened to him.
To them, he simply vanished in the storm.
Right. The police had his remains, but standard DNA testing couldn't find a match in any system.
But in 2025, the evidence was sent to Othram. By December of that year, using genetic genealogy, they finally identified him. He was 77-year-old James Donald Schlake. He was living just a few miles from where his body was found. 8 years of agonizing uncertainty for his family finally resolved by a sequencer.
Case 2 highlights a completely different kind of obstacle. This wasn't just a battle against the elements, it was a battle against international bureaucracy and identity confusion, the case of Igor Kolomiats.
Remains were found in 2014 at an RV estate on Big Pine Key. But when investigators tried to figure out who he was, they walked into an absolute nightmare of immigration records.
It was an identity maze. When they started pulling threads, they found multiple immigrants in the U.S. systems, all sharing the exact same name, Igor Kolomiats.
Oh, geez.
Investigators were paralyzed. They couldn't arbitrarily assign the death to one of these men without definitive proof. If they guessed wrong, they would be telling a family their loved one was dead when he was actually alive somewhere else. So the remains were classified as officially unidentified for over a decade.
It wasn't until April 2025 that they finally untangled it. They used genetic genealogy to narrow down the family line and then combined that with fingerprint records and Homeland Security data. They meticulously eliminated the other man named Igor Kolomiats, who had documented border crossings after the remains were found in 2014. They finally traced it to a 52-year-old man from Ukraine who had last entered the U.S. via a Miami border crossing in October 1997.
His 1997 driver's license photo matched the medical examiner's description. 11 years later, the puzzle was solved.
And the momentum in Monroe County didn't stop there. In March 2026, they announced the resolution of Case 3, Alfonso James Spikes. His remains were found in 1988 in Key Largo. He had been unidentified for an astonishing 38 years before DNA sequencing gave him his name back.
Those are 3 vastly different cases: an environmental accident, an immigration maze, and a nearly 40-year-old cold case, all solved in the same county within months of each other.
But as miraculous as these identifications are, there's a serious blind spot in the system. And our sources highlight a case that is happening right now as we speak to illustrate this limitation. Let's look at the Minneapolis railcar John Doe.
This is a case from 2003. An unidentified man, estimated to be between 30 and 45 years old, was found dead near a rail car in Minneapolis. He had a few distinguishing features: missing teeth, blue eyes, and crucially, a smallpox vaccination scar on his arm.
That scar strongly indicates he was born before the 1970s, when those vaccinations stopped being routine in many parts of the world.
Right. For 23 years, he's just been a John Doe. Recently, his case was taken up by the DNA Doe Project. They are an incredible group of pro bono forensic genealogists who volunteered their time to identify human remains. They built his DNA profile, they uploaded to the databases, but they hit a wall.
And this raises a crucial issue that we have to talk about. Genetic genealogy relies entirely on demographics.
It's a massive database bias. We have to look at who is taking these consumer DNA tests. The vast majority of people who upload their DNA to sites like MyHeritage or Family Tree DNA in the United States are of Western European descent, specifically British and Irish heritage.
Okay.
This is largely because the U.S. is a melting pot, and descendants of Western European immigrants are highly motivated to research their ancestral roots. If your ancestors are from those regions, you are highly likely to have relatives in the database.
But if you are from other parts of the world, your representation drops dramatically. And that's exactly the blind spot the railcar John Doe fell into. The DNA Doe Project discovered that this man is of Polish or Eastern European descent.
And those populations are drastically underrepresented in U.S. genealogy databases. In many parts of Europe, people already know exactly where their great-grandparents are from, so there is less cultural motivation to take consumer DNA tests.
That makes sense.
Plus, different regions have different privacy laws regarding genetic data. All of this results in a demographic skew in the databases.
But despite the lack of direct close matches, the genealogists managed to pull off a minor miracle in April 2026. Through distant, fragmented matches, they confirmed strong genetic ties to a specific town, Borno, in southern Poland.
They even identified a list of specific family surnames linked to his tree, names like Brol, Bereza, Kurczarsik, Polk, and Soa.
They have the region, they have the surnames, but they still don't have his exact identity. The genetic distance is just slightly too far to make a definitive identification without a closer relative in the system.
So right now, there's an active public appeal happening. The DNA Duo Project is practically begging people of Polish descent who have taken consumer DNA tests to upload their raw data to GED Match, DNA Justice, or Family Tree DNA.
Because the math of this is wild. It literally just takes one person, one second cousin, one missing thread, logs on and uploads their file, and this 23-year-old mystery is solved instantly.
It emphasizes that the power of this revolution is collective. The databases only work if the public participates. And that concept of collective participation leads us perfectly into the final major shift in our sources, the global horizon.
Because if regional databases in California and Florida can achieve these results, what happens when the entire globe connects?
Right.
That thought gives me goosebumps. Because the focus of our sources here is clear. We need to understand how new international methods are about to unlock older cases worldwide. Let's start with Interpol's unprecedented international push.
In 2024, Interpol launched a campaign called Identify Me. Historically, Interpol issues what are known as black notices, international alerts regarding unidentified human remains. But these notices were traditionally restricted strictly to law enforcement channels. The public never saw them.
The Identify Me campaign changed that entirely. They unprecedentedly shared extracts from 47 black notices directly with the global public.
These were 47 unidentified women found across 6 different European countries. They released physical descriptions, facial reconstructions, and clothing details, and the campaign is working. The sources highlight a massive success from just May of 2026.
Yes. A teenage girl was found dead in the River Main in Frankfurt, Germany, back in July 2001. She went unidentified for 25 years. But through the Identify Me public campaign, combined with advanced forensic DNA work across borders, she was finally identified.
And the shockwave of that identification led directly to the arrest of a 67-year-old man, her own father.
It is a devastating resolution, but it is a resolution, and there are still 41 women from that campaign waiting to be identified. But what really accelerates this global effort is a scientific breakthrough that was announced in April 2026, which frankly sounds like science fiction.
This is perhaps the most structurally significant scientific advancement since forensic genetic genealogy itself. Reports from The Telegraph and Yahoo News detailed a brand new DNA profiling method developed by scientists affiliated with Interpol.
And the staggering implication here is the age of the evidence they can now test.
Exactly. We discussed earlier how Othram can process degraded evidence from the 70s and 80s. But this new Interpol method pushes the boundary back even further. They can now extract usable genetic data from evidence dating back 70-plus years.
Wow.
We are talking about physical evidence from the 1940s and the 1950s.
I need to pause and emphasize how insane that is. Evidence from the 1940s. It has been sitting in cardboard boxes in basement evidence rooms for almost a century. Every forensic scientist on Earth previously considered that material completely destroyed by time. The DNA molecule should have completely turned to dust.
But now, scientists have the key to unlock it.
And when you combine that new method of extracting ancient DNA with Interpol's iFamilia database, which holds over 32,000 kinship profiles from 82 different countries specifically designed to identify missing persons globally, the implications are profound.
Older cases worldwide, crimes committed during the mid-20th century, are about to be unlocked at a scale we have never seen. International borders and the passage of human lifetime will no longer protect a perpetrator.
Which brings us back home to the United States, because while the global outlook is expanding, the scale of the American backlog is terrifying. Our sources note that the U.S. has an estimated 250,000 unsolved homicides.
250,000 people waiting for justice.
But the mechanisms to address that backlog are accelerating simultaneously. We are seeing states aggressively expanding the criteria for CODIS entry. It used to be that your DNA was only entered if you were convicted of a felony. Now many states have passed legislation allowing DNA to be taken upon arrest.
That simple legislative change is causing the databases to grow exponentially.
And while the databases grow, private labs like Othram and Parabon Nanolabs are industrializing the genetic genealogy process. They are no longer doing this one case at a time as a novelty. They have streamlined the bioinformatics.
They're working through massive backlogs, processing hundreds of cases systematically, day in and day out.
So if we synthesize all of these vectors, expanding arrest databases, consumer genealogy databases growing by millions of users a year, new extraction methods reaching back to the 1940s, and automated international cooperation, we're looking at an exponential acceleration in global cold case resolutions in the coming years.
We really are.
So what does this all mean? As we bring this deep dive to a close, I want to bring the focus intimately back to you, the listener, because it is so easy to get lost in the acronyms, STRs, SMPs, CODIS, FGG. It is easy to view this as a fascinating science experiment, a puzzle to be solved. But behind every single one of those acronyms is a real family.
It is the human cost of the wait.
Exactly. It's Karen Percifield's family waiting 49 agonizing years just to know who took her life. It's Jeannie Child's family waiting 26 years. It's James Schlake's family waiting 8 years, not knowing if he was dead or just lost after a hurricane. It's the family of the Minneapolis railcar John Doe, who right now, at this very second, might not even know he's been missing for 2 decades.
There is a quote synthesized from our sources that captures this perfectly. It notes that DNA doesn't grieve, it doesn't get tired, it doesn't move on with its life, and it doesn't forget. It just waits in the dark until we finally have the science to listen to what it has been trying to tell us.
And we're finally listening. Justice is no longer bound by the limitations of the year a crime was committed. A murder in 1976 can be solved in 2025. The cold cases aren't closed. They're just waiting.
The timeline of justice has been permanently rewritten.
Which leaves me with a final thought I want you to ponder, something that builds on everything we've discussed today. If scientists can now pull usable DNA from 70-year-old evidence, and consumer databases are growing by the millions every single year, are we approaching a mathematical inevitability?
Will we reach a point in our lifetime where the very concept of an unsolved mystery ceases to exist, making it biologically impossible to ever truly disappear?
Thank you for joining us on this deep dive. If this information shifted your perspective, please share it. The more people who understand this science, the closer we get to answering those waiting families. Stay curious, and we'll catch you on the next deep dive.